Welcome to week 2, COVID-19, Health Systems and Pandemics. Once again, I'm Dr. Michael Solzinski, Professor of Microbiology and Immunology at Geisinger Commonwealth School of Medicine. Welcome again to our MBS graduate students, medical students, members of the Geisinger community, and any others who may be joining us to learn about SARS coronavirus and COVID-19. We'll start off with a look back and a look ahead, what we covered last week. Last week we covered COVID-19 as a new disease. We introduced the topic of a zoonotic infection, that is an infection from animals to humans. And we talked about how the SARS coronavirus 2 likely had its origins originally in the bat population, underwent some genetic change that we call antigenic shift to move to some yet unidentified intermediate host, which again underwent another antigenic shift so that the virus is now transmissible to humans as part of its host range. We talked about how the first incidence of disease, pulmonary disease, was described on the very last day of 2019. And we talked about this as a new disease that had been characterized quickly over the coming weeks and months. Next, we talked about how the virus spreads. We talked about person-to-person transmission and the public health recommendations to minimize the risks of infection. We talked about the consequences of infection and said that they varied widely. On one extreme, asymptomatic infection with no symptoms at all, and on the other extreme, severe, sometimes fatal infections. We talked about the nature of viruses. We said that all viruses are made up of two components, a genome and a capsid. Genome consists of either DNA or RNA, but never both. We said that SARS coronavirus 2 was an RNA virus, and as an RNA virus, we said that there was a special tendency of viruses with RNA genomes to mutate and to evolve quickly. We talked about the virus replication cycle. We listed the 6 steps that each virus takes to replicate and make more of itself. And we said that the first step, the attachment step, was arguably the most important, because if attachment takes place, infection takes place. Attachment is a very specific interaction between something on the surface of the virus with something on the surface of the cell. We talked about the specific properties of SARS coronavirus 2. We talked about its shape and its name. We talked about the fact that it has an envelope. And as an enveloped virus, what that meant in terms of its ability to survive outside the body. Focusing further on the envelope, we talked a lot about a particular protein known as the spike glycoprotein, projecting through and extending from the envelope, giving the virus its characteristic crown appearance. We said that the spike glycoprotein was important because it is the molecule that will be recognized by the host receptor. We said that the spike glycoprotein is important because it is the molecule that will be recognized by our immune system as something foreign to the body. And as such, we'll see today that that spike glycoprotein is going to be a key protein for vaccine design. We ended up talking last week about the key discoveries over the past 9 months. Those key discoveries including the determination and publication of the complete genome of SARS coronavirus 2, 30,000 bases of ribonucleic acid in a particular order. And I'll remind you today of how important that breakthrough has been in our ability to do diagnostics as well as vaccines. We talked about the fact that only less than 10 major strains of the virus have been detected circulating through the population. And we said this was remarkable for an RNA virus, which at least has the potential to produce hundreds, if not thousands, of different strains. Last time we said that a key discovery was the ability to cultivate or replicate the virus in tissue culture and living cells in the laboratory. And we talked about the fact that there was a animal model, that is to say, within the host range of this virus, there were specific animals that could be inoculated with purified virus, and after inoculation, those animals would develop clinical disease. Today, working ahead with week 2, we're going to talk about testing, therapy, and vaccine strategies. Testing will include both diagnostics and screening. PCR tests will detect the SARS coronavirus RNA genome. Serological tests to detect antibodies throughout the body made against the virus. We'll talk about drug therapy and convalescent serum using a procedure known as passive immunization. And we'll spend a lot of time talking about vaccine strategies and the plans for a safe and reliable vaccine for global distribution. For those of you who may be enrolled as a student in class, these are the learning objectives for week 2. To explain how PCR is applied as a diagnostic test to detect SARS coronavirus in nasopharyngeal samples. To describe how antibodies produced against the virus are being applied to determine present and past infection, how those antibodies might be produced for vaccination purposes. To summarize the evidence to date on antiviral drug therapies, outline the current strategies for global development of vaccines using criteria of safety and efficacy, and to have an understanding of the technical and practical challenges of production and distribution. Summarize ethical considerations associated with vaccine testing, delivery, and administration. Let's begin with an overview of diagnostics, specifically for COVID-19. By diagnostics, our goal is to detect some marker of infection, such that infected persons have that marker that is detectable, and uninfected persons do not. Well, what is the marker? The marker could be the virus itself. Marker could be parts of the virus, viral RNA, viral proteins. Markers could also be antibodies made by the body in response to the virus or its parts. So the goal for COVID-19 diagnostics is to identify those persons that are infected who have that marker, to medically treat them if appropriate, to isolate them from others who are not infected, and to follow how the disease moves throughout the population. We'll begin with a discussion on polymerase chain reaction, PCR. PCR is a method widely used to make millions of copies of a very small quantity of nucleic acid. So it follows the rule of the proverbial needle in a haystack, so that if a person has a small amount of that particular nucleic acid in their body, the PCR test is an amplification strategy where small quantities of that target become amplified such that there are millions of copies of it, which are much easier to detect. In order to design a PCR test requires the knowledge of the genome. You need to know what the sequence of the genome is, at least partially. And again, that highlights what we talked about in week 1, the importance of establishing what that genome was in terms of the sequence. PCR is considered to be the gold standard of diagnostic tests, the most reliable test in terms of clinical diagnosis. If you look at the illustration on the left, you see the sample being taken as a nasopharyngeal swab. Second part, which we'll go into detail on the next slide, shows how the DNA is amplified in the test tube in the laboratory. And the third part talks about detection and determining whether the sample is positive or negative. So polymerase chain reaction is described as a DNA amplification test. Well, we know that the genome of SARS coronavirus is RNA, so the very first step in this assay is going to be to take the viral RNA and switch it over to a DNA. After that happens, we do the procedure of amplification, which involves heating and cooling the samples in the presence of small pieces of DNA called primers. The primers are specific for the nucleocapsid gene of the SARS coronavirus. So this brings tremendous specificity for the amplification. So it's not going to amplify just any nucleic acid, it's going to amplify specifically the nucleic acid of the virus that we're interested in. So every time the sample is heated and cooled, we call that a cycle of PCR. We typically do the procedure for 20 or 30 cycles, and with each cycle, the amount of amplified DNA doubles. So we get a quick logarithmic increase in the number of copies of a specific nucleic acid. The details then for SARS coronavirus 2 PCR, it's a nasopharyngeal swab obtained from the patient. Swab is then placed in a tube with preservative, sent to the lab. Sample is partially purified, treated to release the viral RNA from the sample. Sample is put in a test tube, and again, as I just mentioned, the very first step is to take the viral RNA and convert it to DNA. That happens using an enzyme called reverse transcriptase. So when we see RT-PCR can refer to reverse transcriptase, where the target is actually RNA that needs to be converted to DNA. Once that conversion takes place, as I said, the DNA is amplified over many cycles of PCR, such that there is an amplification many million-fold. Positive sample is then detected by the shape of a curve caused by fluorescence that is determined and observed on a computer monitor. A little bit of details for the PCR test. There are actually 3 different sets of these primers that are specific. 2 of the primer pairs target the viral N gene or nucleocapsid gene, as we described last week, as being associated with the viral RNA inside the virus. There is a single primer pair that's an additional primer pair that confirms that the sample is amplification competent. In other words, we are detecting a human gene in the sample to verify that the sample is amplifiable. This is really important in validating a negative result. We also have negative controls and positive controls. Positive controls typically used are a non-infectious positive control DNA version of the viral RNA. A flow chart showing you how the test is done. Again, starting with our nasopharyngeal swab. Sample is collected, kept at refrigerated temperature until it makes it to the lab. RNA extraction is step number 3 to partially purify the RNA. Step number 4, our real-time PCR. The little Q in front of the PCR says quantitative. So our test is actually going to be able to tell us how much viral RNA is present in a sample. Our test results, number 5 there, I've actually made this a little bit easier to see on this slide, showing you positive and negative results. Again, we are looking indirectly at amplification by looking at increases in fluorescence, because the more amplification that takes place, the more fluorescence that is determined by a meter inside the instrument. So for a positive test, seen on the left, we see a typical sigmoid curve. sample is exceeding some threshold limit, telling us that it's a positive sample, whereas the negative sample shown on the right does not have that fluorescence and therefore is interpreted as a negative. So the PCR test detects a part of the N gene. Remember, the N gene is a relatively stable gene in the genome. That's why the nucleocapsid gene was chosen as the target here. There are some challenges with PCR as reported nationwide, and the challenges are these. The procedure is labor intensive and expensive with specialized equipment and personnel. Because of the cost and availability of PCR, universal testing is not practical. Sampling is difficult, sometimes misses positive samples, such that symptomatic persons may test negative one day. A retest 24 or 48 hours later may test positive. It could well mean that the sample taken was taken too early, such that there was not enough virus or viral RNA in the specimen to come up as a positive. An issue, particularly early on in the pandemic, was that there was a delay of getting specimens to the lab. And the longer the delay, the greater the chance of sample degradation on the way to the lab. And then finally, another challenge is that the PCR test only detects active infection. So it may miss very early infection if there's not enough virus, and it is not capable of detecting cleared infection in which the virus and the viral RNA is gone from the body. Using PCR as a diagnostic test for SARS coronavirus, just a nice summary: using a newly infected patient during the incubation period. Very early in the incubation period, it could well be that the PCR test is going to be negative. Not enough virus or viral RNA to come up as a positive. A symptomatic patient will likely test positive. However, a recovered patient for which the virus and viral RNA has been cleared, this test is going to be negative. There are going to be some people for which the PCR test is probably not done, and that would be an asymptomatic infected patient, people with mild or uncharacteristic symptoms that we described last week probably would not present themselves for a test. And then we have examples of low-risk symptomatic patients. That would be, for example, young people for which severe disease is not likely. The recommendation may simply be to stay at home so that you're not presenting a risk to others, and in which case the PCR test probably won't be done. So the bottom line of this, based on our PCR testing alone, we are missing some unknown number of people that are infected. Let's switch our discussion to talk about the detection of antibodies against SARS coronavirus 2. Serological testing for antibodies or antigens, is based on this particular theory. Persons who have never been infected with SARS coronavirus 2 will not have antibodies against the virus in their blood, and we say such persons are seronegative. Persons who have been infected, either with symptoms or with an asymptomatic infection, either way, will have antibodies against the virus in their blood, and those persons will be seropositive. There are two classes of antibodies that can give us information whether the infection was a past infection or whether it is an active or very recent infection. So, for example, the presence of IgG antibodies would indicate a past infection as opposed to IgM antibodies in the blood, indicating an infection that is still active or very recent. For most diseases and most pathogens, you can exploit these observations to study how the virus moves throughout the population and how many people had been exposed or infected. Theoretically, antibody testing should be able to determine the presence of antibodies in the population, which we call antibody prevalence. The theory is that IgG antibodies in a person's blood will protect them from future infection if they are exposed to the virus. And it shouldn't matter whether those antibodies are there in response to a symptomatic infection with illness, asymptomatic infection without illness or symptoms, or vaccination, which we don't have today, but the hope is hopefully soon. Nonetheless, the presence of antibodies, we are expecting a protection from future infection. So the presence of antibodies, therefore, not only indicates past exposure to the virus or some viral protein, but at least in the historical view, it also suggests future protection. With antibody testing, there are a number of important challenges as reported nationwide, and those challenges are issues both with test sensitivity and test specificity for the antibody tests. Sensitivity is the ability to detect the presence of antibodies such that an infected person should be testing as positive. That's the test with high sensitivity. Specificity is the ability to confirm the absence of antibodies when they are truly absent. That means an uninfected person should be testing as negative with a good specificity. Does being seropositive protect a person from future infection? Truthfully, the answer is we aren't sure at this point in time. There have been a few rare documented cases where persons have been reinfected with SARS coronavirus 2. Why is this an important question? It's an important question because being seropositive and protecting a person is really the basis of vaccine design. We are intentionally creating a vaccine so that people will seroconvert to be seropositive under the assumption that being seropositive, having those antibodies, would protect you from future infection. In addition to PCR and the antibody tests, there are a few other tests that are worth mentioning. There are COVID-19 antigen tests. This is something that you may not have heard about because it is not in the public press. These are laboratory-based and rapid point-of-care tests, as shown on the right, that detect SARS coronavirus viral proteins or antigens in the patient samples. So the antigen test is for diagnosis of active infections, but not past infection, similar to the way we view the PCR test. However, the COVID-19 antigen tests are not nearly as sensitive or as specific as RT-PCR. If you look at the picture to the right, these are point-of-care tests for COVID-19 antigen. Patient specimen is put into the bottom where the specimen block is. There is a capillary action that draws the specimen to the top. And we see a positive test on the left, negative test on the right. This is similar to a pregnancy test with the same kind of technology, where the presence of two bars on the left, a control, and the test indicates a positive sample, and on the right, a negative sample. If there were no bars, nothing for either C or T, that would indicate an invalid test. There are also tests for the management of COVID-19 patients. So beyond the tests that diagnose or detect the virus or its antibodies, there are other tests that are used in the management of patients with COVID-19, and these are tests to detect biomarkers related to inflammation. And again, I refer to the last class where we talked about the cytokine storm and MAS. So once patients are diagnosed with the COVID-19 disease, these additional tests can be used to inform patient management decisions, particularly in patients that have severe infections. Look for an influenza SARS PCR test called the Flu SC2 multiplex assay coming in the next week. This is an RT-PCR test that both detects and differentiates SARS coronavirus 2, influenza virus A, and influenza B virus from upper and lower respiratory specimens. We call this a multiplex assay because with a single patient specimen, you can test for SARS coronavirus and 2 influenza viruses. The test will tell you whether the samples are positive, and it will differentiate each of those 3 different viruses from each other. Expect this to be very useful in the coming flu season. Let's spend a few minutes talking about antiviral drugs. To date, there are no antiviral drugs that are currently approved for patients with COVID-19. This is not particularly unusual for a virus infection. Traditionally, most viruses do not have effective antiviral therapy. There are some exceptions. Some of the exceptions are hepatitis C, hepatitis B, HIV, and influenza virus. But the fact that we haven't had a very good antiviral therapy yet is really not all that unusual. As you are aware, over the past several months, a number of medications were under investigation as therapies for SARS coronavirus 2 infection, including chloroquine, hydroxychloroquine, either in the presence or absence of the antibiotic azithromycin. Those drugs have not fared very well and are not considered to be useful therapies for patients with SARS coronavirus 2. Still under investigation, antiviral drug remdesivir is an adenosine nucleotide analog and is still being examined with clinical trials. There is one investigational therapy that is showing some signs of promise. That is the drug dexamethasone. In the United Kingdom, with a randomized clinical trial, showed that this steroid drug could reduce COVID-19 related deaths by 1/3 in patients on ventilators. Also, those who were receiving oxygen therapy, not on ventilators, had a 20% less risk of COVID-19 related death. However, for patients without serious symptoms, dexamethasone did not appear to provide any particular benefit. So for those most severe cases, dexamethasone has been shown in clinical trials to be efficacious. There is another experimental treatment using what we call convalescent serum. Convalescent serum means taking a serum from patients that have been infected with SARS coronavirus 2 and have survived the infection such that the virus is no longer in their blood. So the model is these people who have convalescent serum have antibodies in their blood, transfer those antibodies to another person to mitigate their infection. This is a process that we call passive immunization. Early evidence results are mixed for passive immunization using convalescent serum, and it appears that not all antibodies are protective antibodies, and passive immunization works for some patients, but not all. Under development for the next few months, expect to see the creation of laboratory-derived monoclonal antibodies. These are highly purified, highly specific antibodies produced in the laboratory and show at least some potential for the treatment of high-risk patients to mitigate their infection. All right, let's talk about vaccines. And of course, the number one question, when will we have a vaccine? In order to license a new vaccine in the U.S., 2 requirements must be met. The vaccine must be proven to be safe and efficacious. Safety, of course, says that the vaccine can be administered without serious adverse drug reactions, side effects. Efficacy means, is the vaccine effective? Is it efficacious? Does it do what it was designed to do? And that is to prevent infection in people that receive the vaccine. So while lots of vaccines may look good on paper, on the drawing board, vaccine design is very easy, but in the real world take neither safety nor efficacy for granted. Here's a slide that shows from the FDA's perspective how a vaccine is developed, approved, and manufactured. 3 phases in clinical trials. The first phase involves about 20 to 100 volunteers. This is a very simple phase 1 clinical trial asking, is the vaccine safe? Are there any serious side effects? And also to determine what might be the most efficient dose or the safest dose that will still achieve the desired end. Phase 2 of a clinical trial involves several hundred volunteers, asking again what are the most common short-term side effects. This phase 2 clinical trial, it does not take a long time. It will then ask, how are the volunteers' immune systems responding to the vaccine, measured by the amount of antibodies produced by people who received that vaccine. A phase 3 clinical trial involves hundreds or thousands of volunteers. Ask the question, how do people who get the vaccine compare with people who do not get the vaccine? Still ask the question, is the vaccine safe in this clinical trial? Is the vaccine effective? And we'll follow a particular vaccine candidate in phase 3 and talk a little bit more about how phase 3 clinical trials are used to evaluate both safety and efficacy. Let's talk about the main strategies for designing vaccines against COVID-19. One of the main new strategies is shown as number 1 on this slide, messenger RNA or mRNA, encoding a SARS coronavirus 2 gene. That SARS coronavirus gene, of course, is going to be the spike glycoprotein because that is going to be the important antigen. So in making this vaccine design, once again, you need to have the information from the genome, so that genome sequence comes into play again and how important that is. So knowing that part of the genome that encodes the spike glycoprotein, you can then create and manufacture in the laboratory a messenger RNA that codes that gene, which ultimately will produce the viral protein after it's received in the body. We'll talk about this with this 2 specific vaccine candidates in just a minute. The second strategy for vaccine candidate design is to make recombinant SARS coronavirus 2 surface protein, particularly our glycoprotein, a spike glycoprotein, in a vector. That vector would be then expressed such that you get large quantities of spike glycoprotein. This technique has been used for other kinds of vaccines successfully in the past. The third approach is to use a viral vector, which is packaging the SARS coronavirus gene for the spike glycoprotein. And I'll give you a specific example of this, such that this recombinant virus as a vector is then going to deliver the viral protein such that the body will recognize that protein and make antibodies. Number 1 on our list was mRNA vaccine design. This is a quick graphic that shows how that works. The messenger RNA for the spike protein of the coronavirus is injected into the muscle. Vaccine triggers production of the protein itself, and then antibodies specific against it, and therefore we get the production of antibodies against the spike glycoprotein. This approach is said to be very safe. Manufacturers say that this is a safe vaccine because it is cell-free, no infectivity, no virus, not possible to become infected by virus at any step. They say it's easy to design based on the nucleotide sequence of the virus. So we have a synthetic viral messenger RNA. Synthetic means it's actually made by a machine in the laboratory by taking that particular sequence of nucleic acid and then put together as a messenger RNA. Once that is injected into the body, it finds its way into cells. In the cytoplasm, messenger RNA is translated on ribosomes to give protein. So that messenger RNA, the information becomes translated to produce the SARS coronavirus spike protein, which is then presented to the immune system to make antibodies. Let's talk about the first specific vaccine candidate that is currently under clinical trial. The vaccine is called mRNA-1273. So this is a messenger RNA vaccine for spike glycoprotein. It is dispersed and packaged in lipid nanoparticles. In general, messenger RNAs are labile inside the cell, so you need something to deliver the messenger RNA such that it doesn't get broken down. It is difficult to get solid clinical data on how these vaccine candidates are performing in clinical trial. So a lot of the information really comes from public press releases from the companies. This report says that the vaccine candidate, mRNA-1273, induced antibody levels that exceed those in human convalescent phase serum. So the observation here is that by vaccinating people, they actually produced more antibodies than people that were naturally infected. Expected release of this candidate vaccine would be late 2020. It's expected that this might be one of the first ones to be approved. The U.S. government has already purchased at least 1 million doses pending FDA approval. It is interesting to note that the name of the company is Moderna, and the end of the company's name is RNA. So that tells you how they are basing their technology for vaccines. Looking at a second leading vaccine candidate, this one produced by the pharmaceutical company Pfizer. And Pfizer, of course, has had a long history of producing vaccines. They produce a vaccine which is based on mRNA. They call it BNT162, messenger RNA, also for the spike glycoprotein. Pfizer actually set up a system where they had 4 different messenger RNAs and internally pitted them one against the other. And the top candidate was this one here, BNT162. Again, difficult to get hard data on how the Pfizer vaccine is working in clinical trials, but the reports that are available say that in animal studies there have been favorable results. That is, that BNT162 is able to produce antibodies and able to also produce a CD4 plus and CD8 plus response. We say this is a cell-mediated response, which may be a very important component of immune protection. For animal studies, the Pfizer vaccine has also been reported to protect vaccinated animals from disease after viral challenge. That's worth a minute or two of a little bit of background. So for the laboratory animals, last time we talked about the host range and we talked about the ability to do challenge infections in animals. So for vaccine candidates, this becomes an important consideration. In this case, you take your vaccine candidate, healthy animals, of course, you will inoculate your healthy animals with the vaccine, and then wait a while, perhaps a couple of weeks. After that time, come back and you will intentionally try to infect those animals with purified virus. We call this a viral challenge. If the vaccine works, it will not only produce antibodies within that animal, but it will protect the vaccinated animals from developing disease after they have been injected with the virus. And the Pfizer vaccine candidate appears to be able to do that. Another vaccine candidate is AstraZeneca. AstraZeneca has their vaccine called AZD1222. This is a vaccine that is taking a different approach, does not use a messenger RNA. Instead, it is using a non-replicating chimpanzee virus to deliver the spike protein. This is an adenovirus that is not infectious to humans. It is theoretically a very safe vaccine. And the idea, of course, is to target the SARS coronavirus spike protein such that once injected into vaccine recipients, the vaccine recipients then recognize the spike protein as foreign and produce antibodies against it. This also seems to be a leading contender in terms of the ability to get a vaccine licensed. Both the US and European Union governments have already agreed to buy millions of doses of the AstraZeneca vaccine pending approval. Again, difficult to get hard data on the clinical trials. However, the company does reveal that to date they have received favorable results with animal studies, producing high quantities of antibodies, producing both CD4 and CD8 mediated response. We call this a cell-mediated response, and again, that might be very important in making a good vaccine. And AstraZeneca also reports that vaccinated animals with AZD1222 also protects. So vaccinated animals are also protected from disease after a viral challenge. I'll give you a little bit of detail about this vaccine currently in phase 3 clinical trials. There are adult volunteers at 80 sites across the United States to evaluate whether this vaccine candidate can prevent COVID-19. There are 30,000 volunteers in this very large clinical trial. The 30,000 volunteers are randomly assigned to one of two groups. One of them is the investigational vaccine group, and the other group is a placebo group. Participants will receive either 2 injections of the investigational vaccine or 2 injections of a saline placebo, approximately 4 weeks apart. So the study is designed such that there are 2/3 of the study people that are enrolled in the study, 2/3 of them receive the vaccine, and about 10,000 people receive a placebo. It is a blind study such that the persons who are receiving the vaccine and the persons that are administering the vaccine are blind in that they do not know whether the person is getting the actual vaccine or the placebo. So in this phase 3 clinical trial, what you do is administer the vaccine or the placebo, and then you follow all the participants, all 30,000 of them. And you are going to compare the presence of antibodies against SARS coronavirus 2. If the vaccine works, you would expect that those who have received the investigational vaccine as a group would produce antibodies against the virus, whereas those in the placebo group would typically not, unless they were otherwise infected. You would also determine if there is a decreased incidence of disease in those volunteers that receive the vaccine. So this is a little bit different doing a human component to the clinical trial, because for ethical reasons, you can't do a challenge inoculation. We can't have people volunteering for the vaccine candidate and then intentionally try to infect them with the virus. So that is why the animal models become infected. For the human clinical trials, we simply follow them over time and ask the question, as they are naturally exposed to the virus in their environment, will they be protected against disease if they are exposed? So you follow the participants looking for antibodies. You are determining whether there is a decreased incidence in those who receive the vaccine. And of course, in terms of safety, you're also comparing the two groups to see whether there is an increased incidence of adverse drug reactions or side effects. So I've given you just a few samples of what are considered to be the top contenders in this race to get a vaccine licensed. There are many others, well over 100 other entities that are involved in making vaccine candidates worldwide. In addition to what we described, there are other approaches that are also being taken. For example, inactivated or killed vaccines, where the virus is treated with formalin to kill it. This is a standard technique that is proven technology that we use for many of our vaccines currently. There are other approaches that include recombinant vaccines using either an adenovirus or baculovirus vectors. The local company, Sanofi Pasteur, is using a baculovirus vector for its vaccine candidate. There is another approach using what is called self-amplifying RNA vaccines. That is another unproven technology, and many other new approaches. Let's talk about vaccines as the important challenges that we face for any vaccines in terms of their safety and efficacy. First of all, challenges to vaccine safety. And let's frame this in the context of present-day views on vaccines in general, particularly in the US. For mostly unfounded reasons, there is a fear or aversion to vaccines. So that is right off the bat a challenge to vaccine safety. We need to have a vaccine that is going to be proven safe to convince people that this is something that they want to get. So in the US, we have 328 million people. We are envisioning a vaccine for hundreds of millions of them. COVID vaccine will be tested first in animal models and then in human clinical trials to demonstrate vaccine safety. But the concern is that in a hurry to get the vaccine out, in a hurry to get the vaccine licensed, we will have limited clinical trials in which limited numbers of persons are enrolled over a limited period of time. The problem is that adverse drug reactions or side effects may be rare and may not be seen until large numbers of persons receive the vaccine. So 30,000 people in a clinical trial sounds like a large number, but it might not be large enough to be able to detect rare adverse drug reactions. If you have a serious adverse drug reaction, for example, at a rate of 1 in 1,000, if you administer the vaccine to 200 million people, you will expect at a rate of 1 in 1,000 that 200,000 persons will have that adverse drug reaction. So, very important that the vaccine safety be examined very carefully. So there is a valid concern that a rushed, poor vaccine would erode confidence in other vaccines and erode confidence in improved second or third generation vaccines. Remember, the rule for vaccines is the rule for medicine in general. First, do no harm. Challenges to vaccine efficacy. Many of the vaccine candidates we just described rely on unproven technological approaches. That doesn't mean that they won't work. It does mean that their success is more unpredictable. For example, we don't have any licensed vaccines that use messenger RNA technology right now. Will the antibodies that are induced as a result of vaccination protect humans? Will it protect them enough against infection such that they will not have a serious clinical disease? Will the antibodies that are produced at the time of vaccination persist, or will they start to wane after weeks, months, years? We would like those antibodies to persist for the lifetime of the vaccine recipient. Are antibodies enough? Many experts predict that any successful vaccine, in addition to antibodies, must involve T cell responses, CD4 and CD8 T cells, not just the production of antibodies. We know that tests in animal models do not always predict effectiveness in humans. Inducing antibodies against the virus doesn't guarantee protection. Persons most in need of protection, that is, the elderly, are often difficult vaccine recipients because they simply can't respond the same way that younger persons can with a more robust immune system. And finally, sometimes vaccines can actually make a natural infection worse. We call this antibody-dependent enhancement of symptoms. It is rare, but it can happen. Then there are logistical challenges for vaccines, and probably the major logistical challenge is what we call scale-up. That is the manufacturing process such that you're now going from hundreds or thousands of vaccines to now tens of thousands, hundreds of thousands, and millions of doses. That is a big consideration. Another logistical challenge is refrigeration, getting the vaccine under refrigeration from the point of manufacture to the point of administration. You need to have an effective network distribution, not just for the vaccine, but also for associated supplies, needles and syringes. 2 doses means that you need to track people and get them back for a second dose. If there are multiple doses, if there are multiple vaccines, what happens if people don't get 2 doses of the same vaccine? We are not able to predict adverse drug reactions when we combine 2 different vaccines because that will not have been examined in clinical trials. What about record keeping, tracing of vaccine recipients and adverse drug reactions? This may be important. It was important certainly in 1976 when we had an outbreak of swine flu in the U.S., and the adverse drug reaction for that vaccine was an increased risk for Guillain-Barré syndrome. What about informed consent? Who will be liable for adverse drug reactions? What about the cost of vaccine? Who will pay for it? The cost of supplies and cost of administration. And for all of those logistical challenges, multiply everything by 2, because almost all the vaccine candidates that we described so far have 2 dose schedules for complete protection. How do you handle multiple vaccines that might be approved by the FDA? Which one might you recommend? Who gets the vaccine first? Who gets the vaccine last? Do you require people to get it? Will there be a government requirement? Employer requirements, school district requirements for children and teachers? What about significantly better vaccines in the pipeline? Should you wait or get a vaccine now? If you are already vaccinated and a much better vaccine now becomes available, what do you do? So I've tried to give you a nice flavor of what those questions might be, and those questions, of course, will come into greater focus over the coming months. But there are lots of challenges in terms of getting a vaccine licensed and distributed to where it needs to be. So that's it for week 2. Here's our summary. We started today with a nice diagnostic overview, talking about the principle of diagnostics. We talked about the technical application of polymerase chain reaction, defined RT-PCR as reverse transcriptase and real-time PCR. We talked about serological testing for antibodies and the challenges of specificity and sensitivity. We talked about antiviral drugs. We spent a lot of time talking about vaccine design and some of the new innovative approaches for the current leaders in vaccine design, at least in terms of progress in clinical trials. And then we ended up talking about challenges to vaccine safety and efficacy, as well as practical challenges for distribution. So now we're looking forward to week number 3 by Dr. Rima Persad-Klem, faculty at Geisinger Commonwealth School of Medicine, who will be talking about pandemic preparation and response, mitigation versus quarantine, and other strategies. It's been my pleasure to be with you for the first 2 classes of COVID-19, Health Systems and Pandemics. Whether you are a graduate student, medical student, health professional in the Geisinger system, or part of our wider community, I hope you found the first 2 classes informative and useful. I am looking forward to joining you as a participant in the class as we progress in the weeks ahead.